Assalamu alaikum. Today in this lecture we will discuss about the type of fermentation. After the end of this lecture you will be able to know about the fermentation on the basis of respiration, based on mode of operation, based on culture media, based on production. Let's talk about that what is actually fermentation. Word fermentation is derived from Latin word which mean favor, which mean to boil something. But the definition of fermentation is actually fermentation is a metabolic process that converts sugar to acid, gases or alcohol. What is fermentation? Fermentation is the process in which we use microorganism and the microorganism convert sugar to acid, gases or alcohol, and these on the commercial scale. Fermentation occur, the question arises that in which type of microorganism mainly fermentation occur and how we can use microorganism for the commercial scale production of products. It occurs in yeast and bacteria and also in oxygen stirred muscle cell as in the case of lactic acid fermentation. Now first of all we discuss type of fermentation based on respiration. The first one is aerobic fermentation and the second is anaerobic fermentation. What is aerobic fermentation? Aerobic fermentation means when fermentation occurs in the presence of oxygen, for example wine, beer and acetic acid such as vinegar, apple cider vinegar need oxygen in the primary or first stage of fermentation. In the conventional way, the definition of fermentation was that fermentation occurs in the absence of oxygen, but there was a scientist, his name was Hans Krebs. He proposed a phenomena. The phenomena is known as Hans-Krebs phenomena. According to this phenomena, fermentation can also occur in the presence of oxygen. Anaerobic fermentation means when fermentation occurs in the absence of oxygen. There are two major types of anaerobic fermentation: ethanol fermentation and lactic acid fermentation. Both these restore NAD to allow a cell to continue generating ATP through glycolysis. Now, types of fermentation based on the mode of operation, that in which vessels we can operate the fermentation and how we can operate the fermentation. There are three types of fermentations: batch fermentation, feed batch fermentation and continuous fermentation. First of all we discuss what is batch fermentation. Batch fermentation, actually in batch fermentation, fermentation proceeds without the addition of any fresh growth media. You can provide the media and you shouldn't proceed without the addition of any fresh media. In batch fermentation, the sterile fresh growth media is inoculated with appropriate microorganism. It includes in 5 steps: media addition, fermenter sterilization, inoculum addition mean that microorganism addition, fermentation followed to completion, cell harvesting for product isolation. Batch fermentation growth phases in batch fermentation. You know that bacteria in batch fermentation, growth is a result of consumption of nutrient. First of all, bacteria grow in the lag phase. In the lag phase, bacteria prepare itself for division. Then it goes to exponential or lag phase. In this phase, the bacteria multiply in logarithmic way. And when there is deletion of nutrition, the bacteria goes to stationary phase. And after the complete depletion of nutrition, bacteria death occur. Now, what is the advantages of batch fermentation? Reduce risk of contamination or cell mutation as growth period is short. Low cost when compared to continuous process. per the same bioreactor volume. Easier to set up and run the continuous fermentation. Can use organisms that are unstable in continuous fermentation. What are the disadvantages of batch fermentation? Due to environmental changes in fermenter, slow growth rate. The bacteria growth rate becomes slow, nutrient level decline with the time, due to which the bacteria grow slow. Higher the chances of contamination due to potential contact with pathogenic microorganism. Lower productivity level due to time for filling, heating, sterilization, cooling and cleaning of the bioreactor. Feed batch fermentation, it is a modified version of batch fermentation. What we can do here, the substrate is added at different times throughout the fermentation, but no medium is removed until the end of the process. Periodical addition of substrate keep prolonged log and stationary pace of microorganism in fermental. This result in the rapid increase in the biomass. For example, vinegar, amylase production and antibiotic production we can use feed batch fermentation. Now what are the disadvantages of feed batch? High cost and labor, here low production yield during filling, heating, sterilization and cooling. Here steady state cannot maintain like continuous because here cannot maintain specific growth rate. Disadvantages of feed batch fermentation we discuss. Continuous fermentation is actually the fresh media is added to the fermenter continuously is a fixed rate. Spent media is also removed from the fermenter. Both removal and addition are done at the same rate, so the working volume remain constant. Under this condition, addition and removal, the cell remain in logarithmic or exponential phase of growth. Advantages of continuous fermentation: more productivity, more cost efficient, less sensitive to the impact of human error, more continuity of product due to continuous addition of nutrient, less labor required due to automation, quality of product is better than other process due to the maintained steady state in this fermentation. Advantages of continuous fermentation we have discussed. In the next lecture we will discuss the other type of fermentation.